Hello friends, this is Lieutenant Dr. Yasin. Today we are going to discuss about the different biochemical concept. So be ready for the biochemical concept. As you have seen the before videos that we are discuss about the different intro part and different classification. Now we are going to discuss about the different biochemical concept. So this is Lieutenant Dr. Yasin is going to be have I'll give you the brief history about the how is going to be happen the biochemical concepts. What are the concepts are there? So very first we are going to discuss about how the cell history is there, how you can engage the cell history. So cytologically, the study of the cell. What do you mean by cytology? Cytology is nothing but the study of cell, and it is give you in a 1665 the English scientist Robert Hooke will have the history of cells. Now you can see the use of microscopes to examine the cog, that is a plant, and the Hooke called what he saw, that is he called as the cell for the generation, or you can say cell for the history. Now the different generations taking place, and day by day the days are going on, and then the history of the cell is going to be viral. And then Robert Brown discovered the nucleus in 1833, and Matthias Sheldon, the German botanist Matthias Sheldon, is 1838. The all plants are composed of cells. He produced that the theory, and he composed a theory that all plants are produced the cells. The Theodore Shaman, the also in 1838, he discovered that animals were made up of cells. So this three trend is followed by the main scientist, that is a Robert Hooke. Robert Hooke followed by this Robert Brown, Matthew Sheldon, and the tradition. Is going to be give us the cell history. Now you can see after the years and past, the Rudolf Virchow, the 1855, the German physician, that cells is only come from the other cells. He produces that theory, and the statement debunked theory of spontaneous generation has been occurred. Now you can see the cell theory, the combined of the work of Sheldon, Shanwan, and the Virchow make up the modern cell theory. And that modern cell theory states that the all living things are composed of a cell or cells. Cells are the basic unit of life, and the all cells come from the pre-existing. These are the theory. These all three scientists has been made. Now you can see the how it's going to be organized by the internal organization of these cells, which can be consist of the cell membrane, cytoplasma, prokaryotic cell, eukaryotic cells, membrane, and nucleus and organelles. The prokaryotic cell, the basic difference between the two, that is the prokaryotic cells and eukaryotic cells, is prokaryotic cell is a type of cell that does not have a true nucleus and a membrane of bound organelles. And organisms within the domains, bacteria and the archaea, is based on the prokaryotic cells, while all other forms of life are eukaryotic. So this is the basic difference between the prokaryotic and eukaryotic. We can see and how defined is the eukaryotic. Eukaryotic cell contains membrane bound organelles such as the nucleus and mitochondria and the endoplasmic reticulum. So today we are going to organize all this. We are going to study about the in this video organization of the eukaryotic and prokaryotic cell history, and the mitochondria, nucleus, and endoplasmic reticulum organisms. This all we are going to today we are going to learn. Organisms based on the eukaryotic cells is include the protozoa, fungi, plants, animals. These organisms are grouped into the biological domain, that is the eukaryotia. Next, you can see the how you can going to be compare and contrast the prokaryotics and eukaryotics. There will be if you they contrast with each other. The prokaryotics are the having the cell membranes contain DNA, ribosomes, and cytoplasma, whereas the eukaryotics are also having the nucleus, endoplasmic reticulum, the Golgi apparatus, the lysosomes, the vacuoles, the mitochondria, and the cytoskeleton. These are the very first we can study about. We can see the bacteria, how we can see prokaryotic cell structure, that is in the form of bacteria, which consists of capsules, cell wall, cytoplasmic membrane, ribosomes, and you can see the pili and flagella, nucleotide, and cytoplasma. Whereas when you can see the eukaryotic example here, it's anatomy of the animal cell. We can have which also contains the Golgi apparatus, centrioles, perioxomes, lysosomes, mitochondria, endoplasmic reticulum, nucleuses, and so on. So you can see the plant cell also. Here it contains the chloroplast, cell membrane, cell wall, Golgi apparatus, smooth endoplasmic reticulum, rhizomes, that is the free into the rhizomes attached, which is free into the action and attach also rough endoplasmic reticulum also. And now you can compare it with the contrast with the animal cell as well as the plant cell. Their animal cell and plant cell both is contain cell membrane, ribosomes, nucleus, endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles, mitochondria, and cytoskeleton. And only the difference in the animal cell is it contains the centrioles, and the plant cell is contain the cell wall chloroplast. Now you can see the how is internal organization of the cell. Cell contains organelles, cell composed components that performs the specific function for the cell. You can see the A, B, C, D. These are the different cell. The organelles, the plasma membrane is a very important in the cell organelles. The boundary of the cell is called as plasma membrane. The composed of three distinct layers. That is the two layers of the fat, and the one layer is of protein. You can see the structure where is in the yellow. You can see the proteins are going to be act in the red, and the carbohydrate and the glycoprotein. You can see the plasma membrane structure. It's a very important. You can see the hydrophobic regions and the hydrophilic regions. The hydrophilic head is going to be act in the yellow, and the green values you can see the hydrophobic regions. Now it is composed of mainly of lipid bilayer of phospholipid molecules, but with the large numbers of protein molecules. The producting the protruding the through the layers. The two types of proteins occurs, that is the integral proteins, and the protrude the all the way through the membrane, and the peripheral proteins that are attached only to one surface of the membrane and to the penetrate to the all of the way through the okay. Now you can see the also the carbohydrate. The moieties are attached to the protein and the molecules on the outside of the membrane, and the additional protein molecules in the inside as well. Now you can see the structure of the nucleus. Nucleus contains the chromosomes and the chromatin and the nuclear poles and nuclear envelope. It's called the nucleus is the brain of the cell. It's a very important factor of the of the brain. The bomb, the cell, the brain of the cell is nothing but the nucleus and the border of a porous membrane which can be covered with the nuclear envelope, and it also contains the thin fibers of a DNA and protein which called as the chromatin, and the rod-shaped chromosomes are contains a small round of nucleus. You can see the produce it also produces a ribosome and RNA, which is very makes ribosomes. The nuclei is the most cells contain one or more highly staining structures called nuclei, and it's simply an accumulation of a large amounts of RNA and proteins of the types of found in the ribosomes. The nucleus becomes considerably enlarged when the cell is actively synthesizing the proteins. And see the next structure is very important. That's the ribosome structure, which consists of the small non-membrane bound organelles, which contains two subunits, site of protein synthesis, protein a factory of the cell, either free floating or attached to the endoplasmic reticulum. Now you can see the ribosome structure of the ribosome is made up of proteins and RNA. No membrane is acting in the action. Most of the numerous organelles made of nucleus, specifically in the nucleus. What is the function of the ribosomes? They aids in the protein synthesis. It free from ribosomes, make protein used by the cells. Ribosomes are the make proteins for the export to the cells, and the cell type is it's found in the prokaryotic and eukaryotic cells and plants and animal cells as well as into the action. Now you can see the endoplasmic reticulum. You can see the a beautiful green picture because of the the small dots are the ribosomes, and the membranes are there. It's a complex network which can transport the channels. There are the two types. One is the smooth and the rough surface. One smooth is ribosome free, that functions in poisons and detoxification, and the rough contains the ribosomes, and it releases a new made protein from the cell. So it is very important. Now you can see the system which can membrane the channels to saccles, they flatten the vesicles continuously with the outer membrane of the nuclear envelope, which can be a rough side of the endoplasmic reticulum, and the smooth endoplasmic as we have discussed before. It starts with the ribosomes, and on the cytoplasmic side a protein anabolisms and synthesized proteins. It modifies also and process proteins. It adds sugar to the protein, results for the in glycoproteins. The smooth surface of the endoplasmic reticulum is and no ribosomes are present. Synthesis of the lipids, sites of various synthetic process, detoxification and storage, or it forms a transport of vessels also. Now you can see we are coming for the very important part, that is the Golgi apparatus, which can be a serious flattened sacs that modifies the packages, it stores also, and it transfers the materials out of the cells. It works with the ribosomes and endoplasmic reticulum, as you have seen in the figure, red one. And it's it's located in the cytoplasma, which is associated with the endoplasmic reticulum. Its structure, which is stacked flattened membrane sacs, called a cysteine. Function of these is modification, sorting, and transport and manufacturing. How we can modify the proteins in the lipids, receive the endoplasmic reticulum, adding non-protein into the component, that is example carbohydrate, is sorting, packaging, and storage of the proteins and the lipids. It's also transport these materials into the vesicles through the cell, and it's manufactured the sum of the certain macronucleus, the example the hyaluronic acid. You can say into the figures, it is given into the figures. The very important last part is the lysosomes. Lysosomes are recycling center of the cellular debris. It's membrane bound to the organelle containing the variety of enzymes, and its internal pH is almost is a 5, 4.9 to 5. It helps digest food particles inside the outside of this cell. So this is the very, very important part. The next part is also very good. It's a centrioles. It's like a bamboo shape, found only in animal cells. It's paired with the organelles found together near the nucleus at right at the angles each other. Role in building cilia and flagella play a role in a cellular reproduction as well. Now you can see the cytoskeleton, the cell membrane, endoplasmic reticulum, and microtubules are placed together into the form. The microfilaments are there, and ribosomes also there, and which can upper side of the mitochondria. Now you can see how its framework of the cell is contain the small microflames and large microtubules. They support the cells, giving it shape and the help in the movement of its organelles. The fibril proteins of these cells are usually organized into the filaments or tubules. This originate the precursor of the protein molecules synthesized by the ribosomes and into the cytoplasma. The precursors of the molecules then polymerize to form the filaments, example the microtubules. The mitochondria is a double membrane, and its size of the it's produced the high energy. It's a source of energy actually, which compound the ATP, which contains its own DNA and mDNA. It is the size of the abricatum. You can see into the inner membrane and outer membrane, and recess is there, and matrix is also there. Now you can see the ATP is converted to ADP. ADP is 2 ADP is converted to ATP. Glucose, fatty acid is converted to the energy sources. These are the formation of the adenosine triphosphate, that is the ATP into the cell, which showing the most of the ATP is formed into the mitochondria, and ADP, adenosine and diphosphate is a very, very important membrane to the for the system. The vacuole is just sac that helps you in the food digestion. This is the vacuole actually, and which helping to the cell maintain its water, and it's also watering the balance of the watering balance of the system, found mostly in plants and protist. It's smaller in one in animal and cell. You can see the organelle structure, how is going to be structure and function of the kinds. You can see the cell membrane surrounds the cytoplasma and other organelles, which can give you the allows the things to enter the and exit the cell, get rid of a waste. Both plants and animals it's available into the cell wall rigid and surrounds the plant cell, which provides you supports and protection, which is also it's only available in the plants. Nucleus, the houses of chromosomes and DNA of the genetic mode, which can control the center and the tells other the organelles what to do. Cytoplasma, it's available in both plants and cell animal cells. It's gel like cytoplasma is a gel like that fills the cell. It provides a suspension to the organelles so they move around the water. Mitochondria, double membrane organelle with the inner foods, is also available both in plant and animal cells. And mitochondria has a very, very special function is that it converts the glucose molecules into the energy, and that the energy is very important to the system. Chloroplasts, which fill the chlorophyll, contains stacks of discs, which contains the site of the okay, we can see the photosynthesis, and which makes food for a plant cell, which is only available in the plant cells. Vacuole, much larger in the plant cell than animal cell. It's a plant large central and animal in the small. It's store the site of water and the nutrients and the waste. The next is what liposomes. Lysosomes are the small circular and the organelle that contains enzymes, and it digests old cells, parts, ends, and removal of the waste. This is the very best, and it's only available in the animal cell. They are the four basic molecules of the large molecules what we can distribute it. The all living things are made up of four classes, that is the large molecule, the carbohydrate, lipids, and protein, nucleic acid, the macronucleus, and the molecular biology. So this is going to be happen. We are going to study. This is all about the history of the cells and the organelles. In the next lecture, we are going to deal with the different factors that the body molecules. We deal with that, and we explain in the brief in next lecture. So thank you so much for watching, and for giving time for patiently. Thank you so much. God bless you.